Hello class, today I'm here with the digestive system. So the digestive system organs can be divided into two big groups, the alimentary canal and accessory glands. The alimentary canal includes the mouth together with everything we find in the mouth, teeth, tongue and so on, the esophagus, the stomach, small intestine. Small intestine is further subdivided into three parts, and the large intestine, rectum and anus. These are organs of the alimentary canal. The other second group is accessory glands, which includes the salivary glands. There are three pairs of salivary glands around the mouth: the parotid glands, right and left parotid gland, right and left submandibular gland, right and left sublingual salivary gland, and then liver with the gallbladder, pancreas. These are accessory organs. So accessory organs include pancreas, liver with the gallbladder, and salivary glands. So functions of the digestive system. Let's see what the digestive system does. Function of the digestive system includes digestion of food, absorption of food. So what is absorption? Absorption of food is transfer of digested food from the lumen of the alimentary canal into blood vessels. Completely digested food being transferred from the lumen of the alimentary canal into blood vessels, into blood, is absorption. Excretion is practically a elimination of unabsorbed residual, which is practically defecation. The digestion process involves ingestion of food, propulsion of food along the alimentary canal, mechanical digestion like chewing with the teeth and churning in the stomach. That's what mechanical digestion is. Chemical digestion is digestion involving enzymes which digest food items. Absorption, I have already defined absorption, and defecation. These are processes of which happen in the alimentary canal. So regarding peristalsis. Peristalsis is movement of food along the alimentary canal. Movement of food along the alimentary canal is known as peristalsis. Peristalsis involves contraction of the segment of the alimentary canal behind the food bolus. Here is the food bolus, and relaxation of the alimentary canal segment where the food has arrived. So look how this wave of contraction is pushing this food bolus along this segment of the alimentary canal. So this is peristalsis. This is peristalsis, and besides peristalsis is shown segmentation. I want you to compare and contrast peristalsis and segmentation. Segmentation is a process which mainly happens in the small intestine. It is contraction of at several points within the small intestine. The significance or the effect of the segmentation is mixing food with enzymes where chemical digestion takes place. Look how the enzyme and food was separate, then they mix, then they completely blend because of this segmentation process. This is several contraction sites along the segment of the alimentary canal. So I want you to notice peristalsis and segmentation. Peristalsis and segmentation are part of mechanical processes which happen in the digestive system. So how is the digestive system structured? Let's look at a picture which shows us a segment of the alimentary canal. If you take a section through any part in the alimentary canal, this is what you would see. If you take a section through any part of the alimentary canal, this is what you would see. This picture is representative of the structure of the alimentary canal. Look, if we start from the innermost part, you find the lumen here, and the lumen is lined with epithelium. You can see that this is labeled as epithelium. By the way, this whole layer is known as mucosa. Mucosa consists of the innermost lining, the epithelium, and then under it the lamina propria, which is composed of connective tissue, areolar connective tissue, and then a thin layer of smooth muscle, which is known as muscularis mucosae. So these three together, they make the mucosa. That's the innermost layer. And then on top of that, or under it, is submucosa. Submucosa is to say under the mucosa. Submucosa is mostly areolar connective tissue, and in it are embedded blood vessels, lymphatic vessels, nerves, and glands of the alimentary canal, which secrete enzymes. You look at them. So this is submucosa. Above the submucosa, on the outside of the submucosa, are two layers of smooth muscle. The inner one is circularly oriented in a circle around the segment of the alimentary canal. This is known as circular layer. This is longitudinal layer. So this smooth muscle is oriented along the length of the alimentary canal, whereas this one is oriented around the alimentary canal. So you can guess, you can tell that when this circular layer contracts, the lumen of the alimentary canal narrows down, whereas when this longitudinal alimentary canal is contracted, the length of the alimentary canal decreases, it becomes shorter. So this is what you would see more or less in any part of the alimentary canal. So the outermost, above the muscular layer, we find the peritoneum. This is practically peritoneum. Peritoneum is the equivalent of the pleura in the lungs or in the respiratory system, the pericardium of the heart. You remember we said those organs are covered by double layer membrane. The same is true here. And the membrane covering organs of the alimentary canal is known as peritoneum. Here it is labeled as serosa. Serosa consists of inner connective tissue layer and outer epithelial layer, which is peritoneum. So this is peritoneum. Do you see it peeled off? So you notice that this is outermost cover. So this is peritoneum. Peritoneum is a double layer membrane which covers organs of the alimentary canal. I need to show you more about peritoneum. Let me get another picture which gives us which shows us how the alimentary canal covers organs of the digestive system. Let me get that picture. I'm looking for it. Here it is. Look, this is a sagittal section through the abdominal and pelvic cavity, and look at the peritoneum covering organs of the digestive system. By the way, we have here stomach covered here by peritoneum all around. We have here a segment of the large intestine covered by peritoneum, and segments of small intestine also covered by peritoneum. This peritoneum covering directly the organs of the alimentary canal is visceral peritoneum, and there is another layer around the abdominal cavity covering the abdominal cavity on the inside that is known as parietal peritoneum. You remember we saw visceral and parietal pleura, visceral and parietal pericardium. The same is true here, visceral and parietal peritoneum. What is special about peritoneum is peritoneum has a part, as you can see here and here, which attach the segment of the alimentary canal to the back of the abdomen. That part is composed of a double layer peritoneum, and its name is mesentery. Do you see this labeled as mesentery? This one also mesentery, which is attaching the transverse colon to the back of the abdomen. This one is attaching several segments of small intestine to the back of the abdomen. That's mesentery. This is one additional special thing in the peritoneum, which you don't find in the lungs and the heart. And one more special thing about the peritoneum here is, look at this apron-like structure which is hanging from the large intestine and the stomach, and this is known as mesentery. As you can see here, it's labeled as greater omentum. This structure has just protective value. If bullet injury or stab injury happens here, this practically goes and plugs the defect. And here is a minor omentum. Minor omentum, greater omentum, and mesentery are composed of double layered peritoneum. Let's look at parts of the alimentary canal part by part. Let's start with the mouth. Within the mouth, as you know, we find the teeth, tongue, and of course the salivary glands release their product, saliva, into the mouth. So it's obvious the role of the teeth is mechanical digestion, chewing. In addition to knowing that these anterior four are incisors, canions, premolars, molars, and wisdom teeth, you need also to know the structure of a tooth. Here is a picture showing the structure of a tooth. By the way, here I want you to study, when you study, the layers of a tooth. The enamel is the white outermost hard part, which is even harder than bone, and then the dentin, and then the pulp cavity with pulp canal and root canal here. So here is the periodontal ligament, the cementum. You need to study the structure of a tooth, not only the types of teeth, how many teeth in adults have, how many teeth. This is a process by which food is swallowed into the esophagus. It's known as deglutition. This first process is voluntary action, where food bolus is pressed against the roof of the mouth by the tongue and pushed back, and then after that what is continuing is peristalsis practically. Peristalsis, as I explained earlier, is a process by which smooth muscle behind the food bolus contracts and the smooth muscle in front of the food bolus relaxes, allowing the food to be propelled along. So you can see it going into the esophagus, moving around, and going all the way into the stomach. So practically, esophagus is a canal through which food is pushed along. No mechanical digestion, no chemical digestion takes place in the esophagus. So this is to demonstrate the deglutition or swallowing process. Deglutition or swallowing process. So what happens in the stomach? Before we talk about what happens in the stomach, I want to show you a little bit about the structure of the stomach. Let's get a picture which really demonstrates the structure of the stomach. Here is a picture of the stomach. Stomach has the following parts: the fundus, this uppermost part. The body is the most part here, and the pyloric canal. The interior of the stomach, as you can see here, has a shriveled appearance, which is these grooves and prominences are known as rugae of the mucosa. The mystery about this groove is, as the stomach gets distended when it is filled with food, this smoothens out. It gives room for the stomach to distend. So this is known as rugae, this shriveled appearance. These prominences and grooves are known as rugae of the mucosa. And I also want you to notice that the stomach has 3 layers of smooth muscle. This innermost layer is oblique layer, this is a circular layer, and this is a longitudinal layer. When I initially showed you the layers of the representative alimentary canal, there were only 2 layers, right? Longitudinal and circular. But here you see one more additional layer, which is the oblique layer. The reason why stomach has triple layer is because mechanical digestion takes place here in the stomach. Mechanical digestion takes place in the mouth and in the stomach. So stomach needs a 3rd layer of muscle for the mechanical digestion, which happens here. Mechanical digestion happens here in the form of churning, kind of mixing, like someone who makes dough out of flour and water. So that helps mixing of the food material. What happens in the stomach can be described as follows. Let's look for the stomach serves as storage for food. By the way, food can be stored in the stomach for 4 hours. Why do we need to know that food is stored in the stomach for 4 hours? Because if a patient, for example, consumes poison, let's say for suicidal purpose, and if a patient arrives within 4 hours, you can insert a tube into the stomach and suck it out and save a patient. That's why we need to know that food is stored in the stomach for 4 hours. Mechanical digestion, as I already told you, and chemical digestion takes place here. The stomach also produces intrinsic factor. Intrinsic factor is a chemical substance which facilitates absorption of vitamin B12. Vitamin B12 is important in the formation of red blood cells. So a substance which allows absorption of the vitamin B12 is produced by the stomach, and it is known as intrinsic factor. These are the functions of the stomach. Stomach produces an enzyme known as pepsin, which digests protein. Let's have a look at the picture of a gland in the stomach. Glands in the stomach. Here is a picture showing us a gland in the stomach. If you take a piece from the wall of the stomach, do you see this tiny rectangle from the wall of the stomach? And you magnify, this is what you would see. This is showing the gastric gland, the structure of the gastric gland. There are multitude, so we see several of them. If we magnify one of them, this is what we get. Within a gastric gland, there are cells which are known as Schiff cells, these ones, which produce pepsinogen, and pepsinogen is converted into pepsin. I said pepsin digests protein, is an enzyme which digests protein, and hydrochloric acid is produced by these cells, which are known as parietal cells. So Schiff cells produce pepsinogen, which is converted into pepsin under the influence of hydrochloric acid. So gastric glands produce pepsinogen and hydrochloric acid. So digestion of protein starts in the stomach. In addition to chemical digestion with the help of pepsin, Also in the stomach takes place also mechanical digestion. We have seen how the stomach is good for mechanical digestion because it consists of 3 layers of smooth muscle. The final product of mechanical and chemical digestion in the stomach is known as chyme, and it is liquid. Look, chyme being transferred from the stomach into the small intestine. The first part of the small intestine is known as duodenum, and this chyme, C-H-Y-M-E, which is liquefied gastric content, is transferred into the small intestine portion by portion. By the way, this valve is known as pyloric sphincter. It regulates the amount of chyme, C-H-Y-M-E, liquefied product of digestion of food in the stomach, portion by portion, approximately a tablespoonful at a time. After that amount is transferred from the stomach into the duodenum, this valve closes by the influence of enzymes known as cholecystokinin and secretin. Cholecystokinin and secretin are secreted in the duodenum. As soon as a portion, approximately a soup spoonful, is transferred, cholecystokinin and secretin stop secretion and peristalsis within the stomach. That way they stop for additional chyme from being transferred into the duodenum until duodenum manages the only transferred part, the only transferred portion. What does duodenum do with the chyme it received from the stomach? What it does is, first of all, as we have seen, the chyme contains hydrochloric acid. By the way, hydrochloric acid is a strong acid. The stomach has the ability to protect itself from the hydrochloric acid because the inner layer, the inside of the stomach, is lined with simple columnar epithelium and mucus, which protects it from being damaged by the acid. But the duodenum is not as protected, so it uses bicarbonate, which is secreted by the pancreas, which is delivered into the duodenum, to neutralize this acid containing chyme. As soon as this chyme, the liquefied product of digestion in the stomach, arrives in the duodenum, it is mixed with pancreatic juice, which contains bicarbonate, to neutralize the hydrochloric acid. Let me show you a picture showing the arrival of the look. This picture shows this is the pancreas, this is the duodenum. By the way, if we continued, here is stomach. This is where the chyme arrives. This is a portal known as, this portal is known as hepatopancreatic sphincter, into which secretion of the pancreas is released into the duodenum. By the way, duodenum is the first part of the small intestine. Small intestine has 3 parts: duodenum, jejunum, and ileum. So the first part of the small intestine, which is next to the stomach, is duodenum, and it is this part. It is C-shaped. What would come here is the stomach. So as you can see, bile coming from the gallbladder and gastric juice, which comes through the main gastric pancreatic canal, they both open into the duodenum together. So the pancreatic juice contains bicarbonate, which can neutralize the hydrochloric acid which comes from the stomach. So in the small intestine, chemical digestion and absorption takes place in a big way. Before we move on into discussing what happens in the small intestine, I want to ask you how, look, protein, for example, meat is protein, right? How come pepsin, enzyme produced by the stomach which digests meat, protein, does not digest the stomach itself? Here is a way by which pepsin, this is the explanation which can show you why pepsin does not digest the stomach itself or any part of the digestive system. First of all, pepsin is secreted in the form of pepsinogen, which is inactive. It becomes inactive only after it gets into the lumen of the stomach, where it is combined with hydrochloric acid. It is hydrochloric acid which changes the inactive pepsinogen into pepsin. That's one. Second, the inside of the stomach is lined with simple columnar epithelium and mucus. Mucus is thick saliva-like fluid which covers the inside of the stomach. So these are the factors which protect the stomach from being damaged by its own enzyme. By the way, when these factors don't work well, when there is a defect of mucosal or epithelial coverage, then ulcer forms, especially when there is a defect of mucosal coverage. This happens in alcoholics, and that results in gastritis, in gastric ulcer. So this is to explain you how the alimentary canal saves itself from being digested by its own enzyme. So let's move on to the small intestine. What's special about the small intestine? Small intestine is a place where extensive chemical digestion takes place. Extensive chemical digestion and extensive absorption takes place. You can consider the small intestine absorption guru, meaning which a part which is really big on absorption and chemical digestion. Before we move on, I want to demonstrate to you parts of the gallbladder and the canals which lead bile into the duodenum. Each part has a name. Look, this is known as common bile duct. This is hepatic duct. This is known as the cystic duct. The cystic duct brings bile from the gallbladder. The cyst is another word for gallbladder. So they merge and become common bile duct, which opens into the duodenum together with the pancreatic duct. So I want you to notice this structure since we are on this picture. What makes the small intestine a place where a lot of chemical digestion takes place and where a lot of absorption takes place? Let me take you to a picture which would explain that. Let's get a picture of the small intestine which shows all those parts. By the way, this picture is showing you a piece taken from the inside of the small intestine. If we take a piece of tissue from the wall of the small intestine, it would look like this. Look, these finger-like structures are known as villi, singular villus. And these villi, the purpose of this villi is to increase the surface area of the small intestine, the surface area of the small intestine. And then look at this. If you take a piece, you look what you would find: microvilli, which are part of the columnar cell. These finger-like structures plus these microvilli tremendously increase the surface area of the interior of the small intestine. Why is this much surface area needed? For absorption purpose. Absorption, if you remember, I defined as transfer of a digested food from here, from the lumen of the alimentary canal into blood vessels, which are here. Do you see blood vessels here? Here is a capillary, blood capillary. This is transferring into a venule, and this is, by the way, a lymphatic vessel, lymphatic capillary. So transfer of food from the lumen into these blood vessels and lymphatic vessels is known as absorption. By the way, all food items, carbohydrates, fats, protein, and minerals, water, are absorbed into the blood capillaries, whereas only fat is absorbed into the lymphatic capillary, which is known as lacteal. So we have seen this when we studied lymphatic system. I'm repeating here. Fat is absorbed separately into the lymphatic capillaries, which are known as lacteals. The rest of food items are absorbed into blood capillaries. So increased surface area because of these finger-like structures, which are known as villi and microvilli, gives the small intestine a capacity to digest food tremendously. So chemicals which are produced by the pancreas and which are released into the small intestine are chemicals which are produced by the pancreas and released into the small intestine are amylase, lipase, trypsin, and hemotrypsin. Amylase, as we have seen before, digests carbohydrate. Lipase digests fat. Trypsin digests protein. Hemotrypsin also digests protein. So all these enzymes can digest all food items. So digestion in the small intestine is completed like this. By the way, in addition to enzymes coming from the pancreas, there are enzymes which are produced by the brush border here. The reason why this is known as brush border is, does this remind you of brush? That's why they call it brush border. Here at the brush border, there are enzymes which are produced by the brush border and which complete digestion. Let me show you that on the example of the carbohydrate, on the example of carbohydrate digestion. For example, if we take carbohydrate food item like, let's say, starch, can be digested by amylase into, let's say, lactose. So this is not complete digestion, right? It needs further to be digested into glucose, which is the smallest particle of carbohydrates, by enzyme known as lactase. That means starch is a polymer. You remember in the beginning of Bio 233 we studied chemistry and we saw there starch is a polymer and lactose is dimer and glucose is a monomer, meaning single unit, double unit, and multi-unit of carbohydrate. So the digestion of starch into lactose takes place in the intestine with the help of amylase, which comes from the pancreas, and then lactose is further digested into glucose by enzyme which is produced by the brush border, which completes carbohydrate digestion into glucose. It's only after completely being digested that carbohydrate can be absorbed. So lactase is a brush border enzyme. And here I want you to notice the relationship between an enzyme and the substrate or the substance it digests. Look, lactase, as you can see here, digests lactose, right? Look, the first part, this comes from here, from the substance it digests. The rest, ASE, is a suffix which all enzymes have. If you see ASE at the end, and the part in the beginning, or a prefix or root part, which comes from the substance, then this enzyme digests this. I'm giving you a clue by which you would know which enzyme digests which food particle. For example, lipase is an enzyme which digests lipid. This is the same pattern. Lip is the first part of the name of the enzyme, as is common for all enzymes, and this lipase digests lipid. Most of them, not all of them, most of them have this pattern. This pattern, knowing this pattern can help you to remember which enzyme digests which food item. Lipase digests lipid. Lipase digests lipid. Lactase digests lactose. So remember that pattern. Speaking of enzymes themselves are protein. I want you to know that enzymes are themselves protein. In addition to that, enzymes can be reused again and again and again. After digesting some lactose, this lactose can be reused to digest another one. So enzymes are usually reusable. They are not used and throw, but they can be reused again. Okay, so what I want you to remember about small intestine is it is a site, part of the small intestine, part of the alimentary canal where a lot of chemical digestion takes place and a lot of absorption takes place. And the digestion is completed not only by the enzymes which come from the pancreas, but also by enzymes which are produced in the brush border of the small intestine. Now, let's see what the role of the bile which comes from the gallbladder and the liver is. Bile helps digestion of the fat. Bile helps digestion of fat. It comes from the gallbladder. We have seen that picture earlier, and it helps digestion of fat. It does emulsification. It helps the mixing of lipase with fat. Otherwise, they would be separate. If you pour oil into water, they would be separate. They would never mix, right? But bile helps the mixing of lipase and fat, that way facilitating digestion of fat by lipase. This is gallbladder. What is the purpose of the gallbladder? What function does it have? It stores bile. Not only storing, it also concentrates. It takes away the water. It makes bile concentrated. Bile is produced by the liver. I want you to remember that bile is produced by the liver. Let's move on to the large intestine. What happens in the large intestine? The function of the large intestine is water absorption, absorption of vitamins, and by the way, synthesis of vitamins also. Beneficial bacteria which live in the large intestine produce vitamins like vitamin A, vitamin K. They are produced by beneficial bacteria in the large intestine, and they are absorbed there. Storage of feces until it is eliminated, until defecation takes place. Mainly absorption of water takes place in the large intestine. Now let's move on to the liver. What does liver do for us? Oh, liver has a lot of functions, a lot of role for us. Here is a list of functions of the liver. Liver produces, as I already stated, produces bile, which helps digestion of lipase. Liver also regulates the amount of glucose in our blood. When there is excess glucose, liver stores the excess glucose in itself, releasing only the right amount. So it regulates the amount of glucose. When there is too much glucose in our blood, that's diabetes, right? So liver stores excess glucose in the form of glycogen. We will come more into that. Glycogen is a carbohydrates in the form of which glucose or carbohydrate is stored in our liver. Glycogen. Don't mix it with glucagon. Glucagon is an enzyme. Glucagon is an enzyme produced by the pancreas, whereas glycogen is a carbohydrates polymer in the form of which carbohydrates is stored in our body, in liver, in muscle. So liver regulates the amount of glucose in our blood by accumulating or storing the excess glucose in the form of glycogen in our liver. What else does the liver do? Liver also removes excess amino acids. It also removes excess fat and stores. It also stores vitamins. It also produces plasma proteins. Do you remember albumin? When we studied blood, we said one of the proteins found in plasma is albumin, which is the most abundant protein in blood. That's produced by the liver. It's produced by the liver. So liver does that also. Liver also detoxifies harmful substances like alcohol or any other harmful substance. That is why food absorbed in the small intestine is transported by the portal vein into the liver before it is delivered to each and every cell. We have seen that by the merging of inferior and superior mesenteric veins and gastric veins, the merging and the splenic vein. All these veins merge to form the hepatic portal vein, which brings blood into the liver, right? So these inferior and superior mesenteric veins drain the large and small intestine. So freshly absorbed food item is transported into the liver via the hepatic portal vein to the liver. So excess glucose is removed and stored, excess amino acid is removed and stored, excess fat is removed and stored in the liver, and toxic substances are detoxified before they are delivered to parts of our body. That way liver has protective function to our body. Okay, gallbladder, we already talked, it stores food. And we have seen that there are organs which have dual function, meaning which participate in two systems. For example, we saw that the pharynx has role with the digestive system. It also has role with the respiratory system. Here we can see that happening regarding the pancreas. Pancreas has role with the endocrine system, and it also has role with the digestive system. Its role with the digestive system is by producing enzymes. Which are the enzymes which pancreas produces? Pancreatic amylase, trypsin, chymotrypsin, lipase. All these are produced by the pancreas. Pancreas contributes to the digestive system by producing these enzymes and also bicarbonate and delivering it into the small intestine, specifically into the duodenum. On the other hand, pancreas has a role as an endocrine organ by producing insulin. I think you know insulin is produced by the pancreas. And I want to show you a picture of the part of the pancreas which produces enzymes. Those structures, those parts of the pancreas which produce enzymes are known as acinars. They are here. Acinar cells. By the way, if you take a section through the pancreas, this is what you would see. These individual cells are known as acinar cells. This is an artist drawn picture showing acinar cells here, which drain into the pancreatic duct, which merge into the main pancreatic duct and drains into the duodenum. These cells produce those enzymes I listed: amylase, pancreatic amylase, lipase, trypsin, chymotrypsin. And these duct cells, they produce bicarbonate. Bicarbonate is a substance which neutralizes the acidic chyme or the acidic liquid food content which comes from the stomach into the small intestine. Whereas other structures, which are known as isolates, within which there are alpha and beta cells, they produce insulin and glucagon. Glucagon is a hormone which increases the amount of glucose in our blood. When is it secreted? When there is deficiency of glucose in our blood, glucagon is secreted by the pancreas to increase the amount of glucose in our blood. Whereas insulin does the opposite. It decreases the amount of glucose in our blood by promoting entrance of glucose into cells or storage of excess glucose in the liver. That way it decreases the amount of glucose. So I am trying to show you that pancreas has double role. I also would like to show you the histological or the structure of the liver. I have a picture here showing that. Look, if you take a piece of tissue from the liver, this is how it would look like. It shows hexagonal lobules, small parts, small parts. If you take one of the hexagonal parts or lobules and magnify, this is how it would look like. This actually is an artist drawn picture, but this should show you that the structure, the architecture of the liver is so complicated. So when inflammation happens to the liver or infection happens to the liver and this architecture gets distorted, then liver no more functions. And liver, as you have seen, is very important organ. If it doesn't function, it's almost equivalent to death. Look, this is the central vein, which is here, and at each angle of the hexagon you can see a triple vessels. One of them is bile duct, the other one is branch of the portal venule, and the other one is portal artery. So these triple veins, they release, or the blood flows from them into these sinusoids towards the central vein, while passing by all the processes which happen in the liver happen. These are hepatocytes, cells of the liver. These ones are cells of the liver. These are sinusoids shell-like, practically blood vessels through which blood flows, and bile is also formed and released or drained into the bile duct, common bile duct, and into the gallbladder. So this is the architecture of the liver. You can notice how complicated it is, and you can use this picture to study for your lab exam on the digestive system. Let's continue with metabolism. Metabolism is a sum total of all chemical reactions which take place in our body. I'm defining metabolism. Metabolism is the sum total of chemical reactions which take place in our body. Or another way of defining metabolism is what happens to the nutrients which were digested in the digestive system and were delivered to cells by the circulatory system. We saw that food materials or nutrients are digested completely. Complete digestion means, for example, carbohydrates, starch being digested completely into glucose, protein digested completely into amino acid, and for example, fat digested completely into glycerol and fatty acid. This is considered complete digestion. After that, it is absorbed into blood vessels, carried by blood vessels, and delivered to each and every cell. So what do our cells do with the nutrient which was delivered to them after being completely digested? That's what metabolism is. Chemical reactions which take place in our body can be mainly divided into these three categories. Either anabolism, which is a combination of two smaller components into one big compound, or catabolism, breakdown of one big compound into two smaller components, or exchange of components by two compounds, as you can see on this. These are the main types of chemical reactions which can take place in our body. So to see what happens to the nutrients digested and absorbed in the digestive system and then delivered to each and every cell, let's look at the main nutrients. The main nutrients are carbohydrates, protein, and fat. So we are going to see how our body uses these nutrients, how our body uses nutrients and what happens to these nutrients. As you know, carbohydrates is mainly used for energy production purpose, meaning for ATP production. ATP production happens in three ways. I have a picture which shows those three ways by which metabolism of carbohydrates takes in three ways. As I said, carbohydrates is used for ATP production. Glycolysis. Glycolysis takes place in the cytoplasm of a cell. If we agree that this is a cell, and if nucleus here, and if we agree that this is the mitochondria of a cell, so glycolysis happens in the cytoplasm of a cell. So the glycolysis you see here happens here in the cytoplasm, and it happens in the absence of oxygen. The byproduct of glycolysis is pyruvic acid. So pyruvic acid is a substrate for citric acid cycle. So by glycolysis, two molecules of ATP are produced during glycolysis per glucose molecule. Glycolysis is breakdown of glucose. The direct meaning of the word is breakdown of glucose, which results in the formation of 2 ATP. This happens in the cytoplasm in the absence of oxygen. Oxygen is not used in this process. That results in the formation of 2-ATP, and its byproduct is pyruvic acid. Its byproduct is used for another process known as citric acid cycle or Krebs cycle, which takes place in the mitochondria. By the way, this is mitochondria in here. So this process produces additional 2 molecules of ATP. Here oxygen is involved, and the byproduct of this process is used in electron transport chain or oxidative phosphorylation process, which also takes place in the mitochondria, which forms 28 molecules of ATP. So totally 32 molecules of ATP. So from one glucose molecule, if all these 3 reactions take place, 32 molecules of ATP can be formed. So the first one, which takes place in the cytoplasm, not in the mitochondria, without oxygen, is known as glycolysis, breakdown of glucose, which produces 2-ATP, and the byproduct of this process, pyruvic acid, is used in citric acid cycle. If we go into the details of these processes, each one of them has several steps. This has multitude of steps, but in general, the byproduct of glycolysis, which is pyruvic acid, is used as a substrate for this process, which also results in the formation of 2-ATP. And the byproduct of citric acid cycle or Krebs cycle is used for oxidation phosphorylation process, which is most productive method of ATP formation, which produces 28 ATP. These are the processes by which ATP is produced from carbohydrates. By the way, the most commonly used food item or nutrient for ATP production is carbohydrates. But when our body runs out of carbohydrates, it can use fat is the second one which can be used. By the way, from a molecule of fat, more ATP is produced than from a molecule of glucose. And if our body runs out of fat, it can also use protein. It can also use protein. By the way, the whole essence of weight loss through exercise is so our body runs out of carbohydrates and starts using the fat. That's how people lose weight. If they decrease consumption but continue exercising, which requires ATP formation, the fat starts to be used to be burned out to produce ATP. So the point is, our body mainly uses carbohydrates for ATP production, but when it runs out of carbohydrates, it can use fat. When it runs out of fat, it can use protein to produce ATP. But the main purpose of carbohydrate is ATP production. Now, we have more or less discussed the metabolism of carbohydrates. There are some words which I need you to know. We have seen glycolysis, which is breakdown of glucose. Here is also another word, glycogenesis. You remember earlier I told you that glycogen, this word, is a long chain of glucose in the form of which carbohydrates is stored in our liver, in our muscles. So glycogenesis is formation, synthesis of glycogen from individual glucose molecules. Look, genesis is creation, formation. So glycogenesis is formation of glycogen. Glycogenesis is formation of glycogen. Glycogenolysis is the opposite, breakdown of glycogen. Gluconeogenesis is synthesis of glucose from fat and protein. So please get used to these words. They will show up in our note, in our exam. So I want you to get familiar with them. What do we use fat for? Here is a list of uses of fat. Fat can be used to produce ATP, as I told you. When we run out of carbohydrates, we use it for ATP production. It helps to absorb fat-soluble vitamins. There are vitamins which are absorbed, dissolved in fat, which cannot dissolve in water. So that's what is meant here. Some vitamins will be absorbed into blood vessels, being dissolved in fat. They cannot dissolve in water. Phospholipids, which are parts of cell membrane, they are fat. So fat is used to form membranes, plasma membrane of cells. Adipose tissue can be used as a protective cushion. By the way, when there is extra fat, when we bang or hit against something, we may not feel it, right? Skinny people feel pain more when they bang against an object than people with a few extra weight or fat pad. Adipose tissue provides insulation. By the way, the fat under our skin helps us so we don't feel very cold when the atmospheric air is very cold. So it insulates, it prevents heat loss, serving as insulator. Adipose tissue is also a reserve energy storage because it can be used when we need it. If it is stored, we have it there. So when we need it, it can be used. So it's reserve energy storage. Lipoprotein made by the liver transports cholesterol. By the way, I would like you to get familiar to two types of lipoproteins. Lipoprotein means lipid and protein together. Lipoproteins are cholesterols. There are two types of cholesterol. LDL, abbreviated like this, and HDL, high density lipoprotein, low density lipoprotein. By the way, they call this one bad cholesterol, and they call this one good cholesterol. This bad cholesterol transports fat from liver to organs, to blood vessels, that's what makes it bad, because it will create a build-up on the inside of blood vessels, which narrows blood vessels. When this happens to coronary arteries, when this LDL builds up on the inside of coronary arteries, that can result in heart attack. Whereas HDL does the opposite, transports cholesterol from periphery to the liver. So does the opposite, they do the opposite. This takes two individual organs, which makes it bad because it results in narrowing of blood vessels, whereas this one removes from periphery and takes it to the liver, takes cholesterol to the liver. So these are both lipoproteins, low density lipoprotein, high density lipoprotein. They are also fats. Lipogenesis, there is a word here, is formation of lipid or fat from glucose. So when our body uses a lot of fat for ATP formation, that can result in the formation of a lot of ketones, which is a byproduct of fat metabolism, and that can result in acidosis, ketoacidosis. That's what happens to the diabetic patients. Diabetic patients lose carbohydrate with their urine, they lose glucose. So when they run out of carbohydrates, they start using massively their fat for ATP production, and as a result, the byproduct ketone accumulates in their body and they run into acidosis. They call that diabetic ketoacidosis. That's emergency situation, which is handled in the emergency room. Let's see protein metabolism. Protein is mainly used as a building block in our body. Protein is a building block. Where do we find it as a building block? You remember keratin in our skin? That's a component, a molecule, which is a component of the skin, which makes our skin waterproof, abrasion resistance. That's protein. Collagens in connective tissues. actins and myosin in muscle, proteins such as enzymes and hormones. Earlier I was telling you that enzymes are proteins themselves. There are hormones which are enzymes. There are also hormones which are steroids, but there are many hormones which are also proteins. So proteins, in addition to serving as building blocks to build our body, they are also enzymes and hormones. By the way, the monomer of protein is amino acid. I'm saying proteins are formed from combination of amino acids. There are 20 types of amino acids. Some of them can be made in our body, but some of them must be provided with nutrients, with food we consume. Those which must be consumed with food are known as essential amino acids. That's what you see. Essential amino acids are those which are inadequately produced in our liver and must be consumed with food. So amino acids are monomers or small components of protein, and there are 20 of them, and some of them can be made in our body, but some of them, if they are not made or not sufficiently made, they must be absorbed with food. So we have seen the three main components. Now let's see the role of some hormones in metabolism. For example, insulin. You know, insulin lowers the amount of glucose in our blood, right? How does it do it? Insulin secretion is stimulated by increased blood glucose. When there is more glucose in our blood, more insulin is produced by the pancreas. That lowers the amount of glucose by promoting entrance of glucose into our cells. It also promotes storage of glucose in our liver, and promotes synthesis of protein from glucose or glycogen. So by doing this, it lowers. On the contrary, glucagon, which is also formed by synthesized by the pancreas, does the opposite, meaning it increases the amount of glucose in our body. How does it do that? By mobilizing the carbohydrates, glucose, stored in our livers, in our muscles, in the form of glycogen, meaning it promotes glycogenolysis. Do you remember we saw the meaning of the word glycogenolysis? Glycogenolysis is breakdown of glycogen into its components. Components of glycogen are glucose. So glycogen and glucose do the opposite. They oppose each other, but they are formed or produced by the same gland, which is pancreas. Adrenaline. It accelerates metabolic rate. Do you remember we saw this on fight or flight reaction in the sympathetic nervous system? It promotes thyroxine, which is a hormone produced by the gland in our neck, adrenal gland. It also promotes metabolism, meaning it promotes accelerated formation of ATP. So by this we are looking at the role of hormones on metabolism, on chemical reactions. What role our liver has in metabolism? We also already saw that it regulates the amount of glucose in our body by accumulating, storing the excess, or releasing stored glucose when there is shortage of glucose. That's what liver does. Liver stores excess carbohydrate in the form of glycogen. Glycogen is a chain of glucose molecules, in the form of which glucose carbohydrates is stored in our liver, in our muscle. Liver can also make carbohydrates from protein, from fat. That is gluconeogenesis. We have seen that word earlier. Liver can catabolize cholesterol, break down, and secretes it in the bile. We know that liver produces bile. That's the other function of liver, and we saw that bile promotes fat digestion. Liver also produces LDL and HDL. They are produced by the liver. So our liver is very important. We have seen that it detoxifies toxic substances, all of that. We have seen the function of the liver earlier today. There is one more concept, actually two more concepts we need to discuss. Metabolic rate. Metabolic rate. Metabolic rate is the amount of metabolic process which needs to take place to keep us active. The amount of metabolic reaction which takes place, or the amount of energy, the amount of ATP which needs to be produced while we are at rest, sitting down doing nothing but not sleeping, is known as basal metabolic rate, to mean basic metabolic rate. And that constitutes approximately 66 kilocalorie per hour. That's for a 70 kilogram adult sitting down doing nothing at a temperature, at a room temperature, is 66 kilocalorie per hour in air-conditioned room. 70 kilogram adult doing nothing, sitting down. Whereas total metabolic rate is basal metabolic rate plus the amount of energy needed to do any activity. When we work, run, exercise, that is total metabolic rate. By the way, factors which affect metabolic rate are body surface area, which means skinny people require more energy than people with higher weight, with more weight. Age, younger ones need more than older ones. Gender, men need more than women. Hormone, higher hormone like thyroxine promotes faster metabolic rate. So these are the factors which affect metabolic rate. Metabolic rate, to repeat, is the amount of energy needed to keep us alive or at rest or active, when we are active doing some function. How is our body temperature regulated? By the way, the main regulator of our body temperature is hypothalamus, which is part of our brain, right? The midbrain, that hypothalamus is the thermostat of our body temperature. Normally, it keeps our body temperature between 36.5 and 37.5. Temperature above 38 is considered fever, right? So, there are thermoreceptors. You remember we have seen baroreceptors, which monitor pressure, blood pressure. We have seen chemoreceptors, which monitor the concentration of chemical substance. There are thermoreceptors, which regulate body temperature. They report to our hypothalamus, which hypothalamus promotes or accelerates metabolic reactions. Every chemical reaction, as it happens, it has a byproduct heat. As ATP is produced, heat is also, as ATP is produced, heat is also released as a byproduct. So that heat, which is released as a byproduct, keeps our body warm. So how much is released to the atmosphere or how much is kept in our body is regulated by our hypothalamus. By the way, we have seen in Bio 233 the role blood vessels under our skin play in heat exchange. When we feel hot, blood vessels under our skin, they dilate and they exchange heat with the atmosphere. They release heat to the atmosphere to keep us within normal range of temperature. When we feel cold, they constrict and retain heat, so we don't freeze, so we don't lose heat when we feel cold. So this is regulated by dilation and constriction of blood vessels under our skin. Thermoregulation happens under the control of the hypothalamus. By the way, when this regulation fails, we can suffer from hypothermia or heat stroke. Heat exhaustion, heat stroke can happen when we feel extremely hot, when we are exposed to extremely hot weather, and when our regulating mechanism fails. By the way, the mechanism by which we exchange heat with the atmosphere are known as conduction, convection, radiation, and evaporation. By the way, conduction is direct conduction, meaning transfer of heat with the atmospheric air. Radiation is, convection is the flow of warm air upwards and cold air downwards. That circulation is known as convection. Radiation is like how the light ray from the sun comes to us, how light ray from a lamp comes to us. That's a convection. Evaporation is the change of liquid water into gaseous states, into vapor. So those are the means by which heat exchange takes place between our body surface area and the surrounding atmosphere. By the way, what is evaporating, what is changing from liquid into gas? Sweat from the surface of our body. You remember that we sweat when we feel hot, and that sweat evaporates, taking heat with itself. The last concept we need to discuss is body mass index. When you went to the hospital, after the nurse takes your weight and your height, they calculate your body mass index, which is practically a ratio of your weight to your height. That is important data because it tells whether we are obese or underweight. So the way to calculate it is if you take weight in kilogram and height in meter, then you divide the weight in kilogram divided by the height in meter squared. That gives BMI. So if that BMI is less than 18, so such a person is underweight. If it is between 18 and 25, that's normal. Above 25, that's considered overweight. If it is more than 30, then that's considered obesity. Above 40, they call it morbid obesity. So this is an equation which helps us to find out our body weight in terms of our height. So this is what I wanted to present to you. So I will see you next time with some discussion on urinary system. Thank you for listening.